right, good afternoon, everybody. It is 1 o'clock Eastern, and so we're about to begin the webinar. Thank you for joining us. It's a spring-like day here in Ithaca. It is like 64 degrees here right now, so this is beekeeping weather. So thank you for joining us. We have a lot of people registered for this webinar. We have over 400 people registered. And so we're going to keep everybody on mute, but there is plenty of opportunity for asking questions. And I will go through and share with you about that. So let me, so first thing to let you know is there's going to be a recording of this webinar. And there's actually, this is, in case you don't know, this is one of a series of 4. This is the first one. And all 4 of them are going to be recorded. And each one will take about a week for us to get the recording edited and up, and it will be available on this link. But also, we will, if you're registered, I'll send you a copy of the link to the recording when it's done, and it'll take some time, hopefully this week, so you should hear by early next week with a copy of the recording. And we welcome your questions at any time. And I ask that you use the Q&A feature because chat doesn't work when we have this number of people on a call. So if you scroll your mouse over your screen, you'll see that there's a black bar that appears. And in the middle of that black bar, there's like a little thing that looks like a file with a Q in the middle of it. And it says Q&A. If you click on there, you can write a question, and you can also put them in there anonymously. So if you have a question you don't want to know people you're asking, you can just click the anonymous option and the questions go in there. And what can happen is we'll be monitoring the questions, and we can answer some of them in writing. We may answer some of them live, but you'll be able to keep track of them too, where you'll see open questions and answered questions, and those ones that are dismissed. It helps us to keep track of everything. And, you know, we may get 3 people asking the same question, in which case we'll answer one and dismiss the other 2, and you'll see, and so that you'll be able to see the log, and it'll be really easy to be able to see your answers rather than chat, which can get really complicated when there's a lot of people. So I should mention, by the way, this is being sponsored by the Northeastern IPM Center, and we receive funding from USDA NIFR that allows this to be possible. And our 2 speakers will mention their organizations and where they receive funding from too. And just know that it's your tax dollars at work going into this webinar. Okay, and so then the next is I'm going to introduce our wonderful presenters. You are in for such a treat. So they are extremely good presenters. You'll really enjoy this. Dr. Kim Skirm is the Chief Apiary Inspector and Apiary Program Coordinator at the Massachusetts Department of Agricultural Resources. Dr. Skirm is a scientist by training and a hobby beekeeper. Having experience in academia, industry, and now government, Kim has always been driven by an intense love of bees to serve in roles supporting bee pollinators informed by the latest scientific research. And Jennifer Lund is the Maine State Apiarist and has over 20 years of entomological experience. Jen has worked on many honeybee projects, including a national colony collapse disorder study, honeybee colony health comparisons of top bar and Langstroth hives, integrated Varroa Mite control effectiveness, the role of honeybees as vectors of blueberry disease, sublethal effects of colonies on low-level pesticide exposure, and health of migratory hives arriving in the state of Maine for blueberry pollination. Jennifer also has several of her own hives that she maintains on her farm in Maine. So as you can tell, we have 2 amazingly experienced presenters with a wealth of knowledge, which they're going to share with you over the next couple of months. And before we begin, so I should say, welcome Kim, welcome Jen, and I'm glad to have you here. And today, actually, Jennifer's going to be doing a lot of the presenting, and Kim's going to be in the background answering your questions, because there's going to be quite a few questions coming in. And we have some questions for you before we begin. There's actually just 3 to begin, and it just gives us a sense of your level of experience and knowledge and who we're speaking with, so that we can tailor the presentation to you. So you should see a poll on your screen right now, And I'm just going to be quiet for a minute or two to give you a chance to answer the questions. All right, I'm going to end the poll and I can share the results with you. All right, so it looks like the vast majority of people are somewhat knowledgeable or moderately knowledgeable, so that's great. And we've got a lot of people in the middle of the road. And most of you have seen a Varroa mite. For those of you who have not seen a Varroa mite, you will definitely see one during this webinar, so don't worry about that. And then some common signs of mite damage. And I will let Kim and Jen answer give more information about the correct choices on that one, and we'll do that at the end. And we have actually at the end of this webinar some questions too, so just so we can gauge increase in knowledge. So given that, let us dive in to the content for today, and I will let you take it away, Jennifer.
All right, can everybody hear me?
Yep.
Yes? Okay,
yes.
Great. So thank you very much. We're really excited to host this. And so I want to thank the IPM Center, the Northeast IPM Center, for hosting this for us. We think it's a really important area for bee health. Of course, Varroa mites is a big deal. So today we're going to go over some honeybee health issues, both in Maine and Massachusetts. We're going to talk about biology, viruses, seasonal growth, and how to diagnose a death to Varroa mites. And then we're going to talk a little bit about the future webinars and some other stuff that's going on in New England over the next couple months that you might be interested in participating in. So next slide. Oh, for some reason we're out of order. There we go. So over the last 3 years in Maine, those of you that are in Maine, probably this is no surprise to you, but we've had losses about 43 to 45% over the last 3 years. And those losses are spread out among the counties. Some do worse than others, and primarily most of our losses are in the winter. Next slide. And really one of the largest causes of death in Maine is Varroa mites and viruses. In 2018-19 survey, this was beekeeper reported losses. Varroa mites and viruses were responsible or reported to be responsible for 26.7% of the winter losses and 8.6% of the summer losses. And I suspect that a lot of the unknown is also attributed to Varroa mites and viruses as well. So next slide. And it's very similar in Massachusetts. Their losses, this is again a beekeeper reported survey, their losses are at 33% this past 2018-2019 season. And Varroa mites was the #1 recorded cause of losses. So go ahead, next slide. And when we go to a national survey, this is also beekeeper reported. Losses reported in Massachusetts and Maine are around that 40% again. And primarily it's winter losses. And Massachusetts, I'm gonna gloat for a little bit, they have the highest losses in New England. I can see Kim laughing. We're the third highest in the Northeast, so just putting it out there. Okay, next slide. And then when we look at what we see, both Kim and I in the field, the inspector reported causes of death. So both of us do hive autopsies. If a beekeeper doesn't know what caused their hive, we go out and examine them. And I would say about 25% falls into the category of queen loss, starvation, poor winter. And a poor winter is not a cold winter. It's one where we have fluctuations in temperature, where we drop 30 or 40 degrees in a couple of hours, that the bees just can't seem to get back together, and that's not great for them. But 70% of that, those losses that we do, or that those surveys that we do, and when we do hive autopsy, 70% of those losses are due to Varroa mites and viruses. And then all the scary things out there that we hear a lot about is about 5%. So that's everything else, including the foul breeds, nosema, vertebrates, insect pests, all of those things. So next slide. And this also goes out, this is Massachusetts, and you can see the counties along the bottom. This again is also dead out evaluations by the inspectors there, and we're seeing really high Varroa mite counts. So this yellow line is a little bit high, but the threshold for treatment, see Berkshire County there, Berkshire County, right above that should be the yellow line for treatment. So you can see a lot of them, a lot of the dead outs have Varroa mite numbers that are really, really high. So next slide.
Maybe I should just say here, if you have a question, if you put it in the Q&A feature, we'll stop and take some questions as we move along. So the first one that we're seeing here is from Tony Braithwaite. He says, when using a drone frame, I remove them at the appropriate time and freeze them. How do I reuse those frames full of dead drone brood and dead Varroa mites, put them back into a hive and let the bees clean them out?
Oh, I think I'm in the hot seat for questions.
Yeah, there you go.
Yes, so thanks so much for joining us, everybody. Yeah, drone frames, we're actually going to spend a little bit of time in future webinars on this, but the short, quick answer is that if you use a wax or a honey uncapper, looks kind of like a little fork that you're using to extract honey to open up those pupal cells and get out as many drones as possible. You know, do the best you can. The bees will clean up whatever is left behind. But, you know, yeah, like you're writing in the question, if you just put a whole frame full of dead frozen drones that are uncapped, they're just going to leave that. But yeah, it would be nice for them to have a little bit more of a clean material to work with. So just uncap as many as you can and try to remove as many drones as you can out of there. Once you uncap them, if you take the frame and hit it a little harder on one side and then reverse it and do it on the other side, those pupae should come out fairly easily, especially if you let them fall out for just a little bit. Jen's shaking her head. Am I missing anything? It's hard to do that without showing somebody with a frame. So, but yeah, it's a good question though. We'll have several photos of that. So if you have issues, hold tight. We're going to talk about these things in good detail coming up.
Great. So feel free to add questions anytime to the, and there's one more question that we can do at this point from Meg Garrison. She said, I'm really looking forward to this series and I'm just getting bees in April. Are there any medications I should have on hand initially? And we may cover that in this webinar, but if not, is there anything to add to that question?
I think the best thing to think about when you're getting medications for bees is that first off, the things we're going to talk about as far as managing for Varroa mites, those are all going to be pesticides. And we're going to talk in good detail about the difference between pesticides that are used for managing Varroa and medications that you can purchase for your bees and use them to treat other pathogens or other issues. But yeah, I think in general, it's a good option to have a couple of different, it's a good idea to have a couple of different options before you start the season. And we'll definitely talk about all the options that are available and how to create a really nice plan for your bees, especially if you're getting them in April. You have plenty of time between now and when we do the future webinars to not only figure out what you need to purchase, but also get a plan that'll help you respond if those things are not working when you're using them. So I definitely don't want to put your question off. I think it's really important, but I also think that we can do a good job of folding that into our additional presentations and really go over that well, because I have a feeling a lot of people are probably saying the same thing. Oh my goodness, how do I start off on the best possible foot with my bees, or tarsi, I guess. So yeah.
Yeah, and this is great timing for you, Meg. It's like perfect timing for you to have this webinar series right as you're getting going. So, all right, well, let's move on to the next section and have Jennifer continue there.
Great. So this is my favorite part. I love talking about mites. Any of my beekeepers that are in the state can attest to that it's my #1 topic to talk about. And so Varroa mites, when we talk about Varroa mites, we typically are talking about Varroa destructor. And this is a small red-brown triangular mite that was discovered in the United States in the late 80s. And it's originally from Southeast Asia, and it jumped off of another species of honeybee onto ours. And so what it does is it feeds off the body fluids of adult pupal and larval bees. And what it is feeding on primarily is the fat bodies. And it's one of the largest ectoparasite to host relationships. And what that means is that it's very large compared to its host. So normally at this point, I hold up this big stuffed mite and show you in relationship what that looks like. But instead, I have a beautiful picture, next slide, showing that's about the size that this mite would be on us. So if bees were the size of us, that's how big that mite would be on them. And these mites can, these bees can have 4, 5, 6 of these on them at any given time. So next slide. And you can see here in the top left is a larva, and you can see all of those mites on them. I think we counted 10 on there. And then down in the bottom, that might be an exaggerated photo, but there are, when mites get really high in a colony, there can be lots and lots of them on top of an adult bee as well. So next slide. So mites have two phases to their life cycle. The first is the dispersal phase, and this used to be called the phoretic phase, but it was actually misnamed, and so now we call it the dispersal phase. And this is where female mites, and this is where the mites are on the adult bees. Female mites pass from bee to bee as they walk past one another. So as two bees approach a flower and they're both feeding on it, one has mites, one doesn't, those mites can just walk right across onto the other bee as those bees touch each other. And they can move to uninfested colonies by drift. So if you have a colony that's relatively free of mites, other bees can come in with their mites and bring them into your colony. You may have read in your bee textbooks that bees do not intermix in colonies. So if you have a bee from colony A and it goes into colony B, they would fight until their death and not let them in. That's completely false. Bees are very social. They're just like us. They constantly go visiting other hives. And especially when there's a lot of nectar and pollen resources out there, bees will go out, collect that nectar and pollen, and come back. And if you have a series of hives, and they'll land on one of those hives, it doesn't really matter which one, especially if they're weighted down with food, and go to the front door, go to the entrance of the hive, meet the bees there, and if they have food, they'll let them in. I always joke with my beekeepers that it's very similar to my house. If you showed up at my house with a cake, even if I don't know who you are, I would still let you in because you brought food, and it's the same with bees. And I also usually go further that you could have the Jason mask and a knife, and I'd be like, oh, you brought cake, come on in, you brought a knife to cut it, great. And that bees are very much the same. If there is food, they will let you in. So what the mites are doing at this point is they're feeding between the abdominal segments on the underside of the adult bee. You can see in the top picture, the mites tucked in between the abdominal segments, and they're up there feeding on the fat bodies of the bees. And those fat bodies for us, our fat bodies are probably not all that important or useful, maybe keeping us warm in the winter and making us more snuggly. But besides that, they don't really serve very much purpose. In insects, though, fat bodies are extremely important. They have 9, 10, 11 different functions. And some of those include if the bee eats a toxic substance, the fat bodies will actually break down that substance into non-toxic components. So they detoxify just like our liver does, the bee bodies. They also serve the function as the immune system of the bee. So if a bacteria or pathogen comes into the insect, they will remove it, kill it, take care of it. And then also fat bodies in insects can be broken down and used for excess energy if they're during times where there's not enough forage or they're being resource starved. And so fat bodies are extremely important in insects, and what these mites are doing is they're actually sticking their mouth parts through the exoskeleton of the bee, regurgitating into the bee, so they're throwing up into the bee, and in their saliva is enzymes that break down the fat body and make it more liquefied. And then the mites will slurp up that liquefied fat body like the perfect milkshake. And so occasionally you'll see pictures or you'll find bees with mites on the thorax. So that bottom picture shows you a mite on the back of the bee. That's a mite that's looking for a new host. And so that one is looking to disperse. The vast majority are going to be feeding underneath your adult bees or in your brood. If there's no brood, these mites can live feeding on adult bees for 5 to 6 months. So during the winter, when we have no brood and the bees are in tight cluster, the mites are happy and feeding on the underside of your adult bees. Next slide. So in their second phase, their reproductive phase, the female mite will leave the adult bee and enter an about to be capped cell. That cell with a larvae, the mite will crawl down to the bottom, hide in the food, in the brood food, and wait for the cell to be capped. Once the cell is capped, she will lay her first egg, which is unfertilized, that develops into a male mite. Subsequent eggs after that are fertilized and develop into females. Right before the adult bee emerges, the mites will mate with one another. So male mites will mate with female mites. And then they will emerge already ready to start laying new eggs. The average founder's mite will produce 1 to 2 offspring per worker cell and 2 to 3 offspring per drone cell. So the reason for that is not to do with the size of the cell. Often when I ask that question in classes, people will say that the drone cells are larger so they can produce more. It's actually the length of development. So because drones take a little bit longer to mature, the mites can make one more, sometimes squeeze out one more offspring during that time. And we find that because of this, mites preferentially select laying in drone cells. So they're 8 to 10 times more infested than worker cells because the mites can produce more in them. And so about somewhere between 50 to 90% of Varroa in a hive can be found in the capped brood cells. So I usually tell beekeepers, if you're seeing a lot of mites on the tops of your bees, ones that are looking for new hosts, you're seeing a very, very small percentage and you have a very serious problem in your hive. And on average in New England, we find that mite populations double every month or so. It's every 3 to 4 weeks that they're doubling. Next. And so we find that Varroa, when it's feeding on bees, it shortens the worker lifespan. It impairs foraging ability, flight behavior, which includes flight behavior and orientation. It increases worker drift and robbing. It increases queen supersedure. It reduces sperm production in drones. It reduces colony level honey production. It reduces colony level winter survival, and it increases the likelihood of virus transmission. So that's just the Varroa Parasitism Effects. So next slide.
So bad news bears.
Yes, it's definitely bad news bears for bees.
Sorry, I had to interject. As I was listening to this, I was like, oh my goodness, with all the coronavirus stuff going on, I feel like this is like the bee apocalypse is having Varroa mites. Yes, it is.
The bee-pocalypse.
Bee-pocalypse. Sorry.
So do you have any questions for us?
There's a lot of questions, a lot of good questions. Folks are really excited here. I'm frantically answering, but I don't do nearly as good as Jen does. She's so much better with these because I type a lot of things. So one that's on here, let's see. We did a medication one. This one is, if I'm waiting, oh wait, go back up. Have you compared the accuracy of an alcohol wash versus the sugar roll method for testing? That's a good question for Jen because I know you've done a little bit of this work with your colleagues down in the Carolinas.
Yeah, so I find that for the vast majority of beekeepers, the alcohol wash is an easier, more effective way to test for Varroa mites. Mostly because with the sugar roll, there's more issues with doing it wrong. I guess that if that makes sense. So it's more likely to not be done in a way that will get you the mite, show you where the mites are. But we're going to talk a lot about that next webinar. So we're going to talk about sampling at that point. But I see a question here from Tony that actually fits in perfectly. If I'm waiting to see mites on adult bees, am I not waiting far, far too long? Yes, absolutely. Yes, without a doubt. I can tell you that when I open up a hive and I see mites on the backs of bees, quite a few of them, if I can pick them out, that hive is going to start showing symptoms of viruses, which we're going to luckily talk about next.
All right. So maybe we'll take one more question and then we'll move along. Have you an opinion on sampling and or then treating any feral swarms or colonies that you collect, just as you would a new package of bees from a producer?
Yeah, usually I tell people when they're collecting swarms or feral colonies to treat them exactly like you would a package. So if you do a mite roll on them and see where your bee numbers are at, your mite numbers are at, and then treat accordingly. So, yep, absolutely.
Great. So let's move along then to the next section on Varroa mite viruses.
All right. So what we got is Varroa mite viruses. Go ahead. Next slide. So there have been more than 20 viruses of honeybees that have been identified, and they spread all different ways. They can spread through mating between a queen and a drone, horizontally between workers when they are regurgitating to each other, doing whatever that thing is, and also vertically from adult bees to young that they're feeding. Most persist naturally in colonies at low levels with no symptoms. So just like us, bees have viruses that are in their systems, but because they're relatively healthy, they don't get sick. It's when they become immune compromised or some sort of stress happens on them that they start showing symptoms. Most of the symptoms are generic, and many have Varroa mite associations. Not all viruses, but many of them do. So next slide. So there's no treatment for viruses. Maintaining healthy, strong colonies and controlling your Varroa mites are the two things that you can do to reduce your likelihood of having viral problems. So one that you may see in your hives, and this one's really common when you have a high Varroa mite counts. It's found worldwide. It was first discovered in the early 90s. And there are several covariants, so it doesn't always present the same way, but there's several different kind of types or strains of the foreign-born virus out there. And it's way worse with Varroa. If you have just the virus itself, it's something that the bees struggle a little bit with, but once you add Varroa to it, it makes it that much worse. And what the symptoms are to this are you have twisted and wrinkled wings. You'll often find bees crawling on the ground in front of the hive. So these bees, because they cannot fly, will emerge from the cell, drop down to the to the bottom board, and crawl out of the front of the hive and kind of mill around in the grass or right in front of the hive, and then eventually they'll die in about 3 days. So you'll see small discolored abdomens, and again, those twisted wings. When I see this in a hive, I can tell you your Varroa mite counts are gonna be very, very high. So next slide. And then as far as all the other viruses, you can't really tell what they are by just the sites of the bees. A lot of the other symptoms are kind of generic. So you have trembling where the bees are kind of shaking. I have a dog that is part Chihuahua, and she is always trembling a little bit. It's kind of like that, so I call it the Chihuahua tremble. Paralysis where the bees are still alive but not moving, or they're moving in a kind of jerky method. Darkened bodies, so this bottom left picture, that black organism right there in the middle is actually a bee, is a honeybee, and so you can get these darkened bodies with some of the viruses. Next to that one on the right, you see some greasy looking bees, so these are actually honeybees too, not wasps, and they're also hairless. The virus is, one of the symptoms is that the hairs do not develop or are pulled off of the bees. You have up on the top right deformities in the wings, and you also have small size or reduced size, and then these bees do not live as long, so that you have decreased longevity. So next slide. So Varroa mites are considered one of the biggest problems facing honeybees. They carry and spread viruses, and the way that these viruses move throughout infected bees is really important to kind of get a grasp of how problematic that they can be. And so what happens is, remember, your bees are feeding on the fat bodies of your mites are feeding on the fat bodies of your bee. And what they're doing is they're actually regurgitating into your bee, and in that, they're digesting the fat body and slurping it back out. Along with the enzymes that are in that regurgitant is also viruses. And so when they're throwing up into your bees, they're also introducing viruses. Those viruses travel and multiply throughout the bee, eventually ending up in this gland called the hypopharyngeal gland in the head of the bee. And that gland is really important because it produces a substance that bees are mixing with pollen to make bee bread. So bee bread, of course, is fed to all the young. So if you have a bee that is infected with viruses, its hypopharyngeal gland is infected with viruses, that liquid that it produces mixes with its pollen to make bee bread is infected with viruses, and then they feed it to their young, who now become infected with viruses. So you can see how problematic these viruses can be in the hives. If left unchecked, Varroa mite will absolutely kill your hive. And it's most problematic in New England two times of the year. Number one is when bee numbers are reduced in preparation for winter. And the second is if we have poor or late spring, summer forage. So next slide. Oh, nope, I guess we're doing questions.
There's actually quite a bit of them here.
Oh gosh, okay.
Here we go. Are you ready? All right. Yep. It says, there's a comment, slide 1. It says that someone just wants to update us that the Varroa mite do not feed on hemolymph. And I thought that was a good question for you, Jen, because I do think that paper, kind of the resounding theme is they only feed on fat body. And my takeaway from the paper was that they do feed on hemolymph, but it's not nearly as involved as what they feed on the fat body. So that's a good point.
Yeah. So I think that first slide, I have it on there, is that they're feeding on the body fluids instead of, because they do feed a little bit on the hemolymph, but primarily they are feeding on those fat bodies, which is really important because those fat bodies are part of the detoxification and also the immune system of bees.
Yeah. I think it's a good point, because I know that's kind of the theme now as we change science with the fat body research, which is awesome. It's fantastic. It really did change. But I think we also, they do feed on that hemolymph. So that's a good point.
It's minor. They can't actually reproduce effectively on just hemolymph. They absolutely need the fat bodies, and they prefer the fat bodies, and that's primarily what they're feeding on.
Yeah. So it's like junk food if you're a teenager versus, eating healthy. You prefer fast food versus, vegetables. So let's see, how long will a mite live without a host or without a hive?
Oh, I don't know.
That's a good question. I've got a fun case where I accidentally collected some bees that we were going to sample, left them in my state vehicle, in the dead of summer and mites came off of those bees and lived in a sealed container for about 3 weeks with no host, so to speak, alive and no oxygen. So although that is very unscientific of an answer, it has no data to back it up except one time. But it's a really good question. We tend to tell beekeepers, if your hive dies in the winter, there's a couple great things that come out of that, once you get past obviously the loss, which is horrible, is that if your bees are dead, the mites are dead. So you have a really nice way to start over. And unless there's something really kind of gaping as far as diseases that are prevalent, you also can get a fresh start for new bees. So you have potentially a lot of comb that's been drawn out that can be used and will give a new colony a boost. But the living mites, that's a good question. I think we need a little bit more information about how long they can live without a host. So let's see. How long is the foundress mite outside of the brood cell before they go back in another cell? Jen's looking at me. Take this question. Yeah.
I don't actually know if we know that either.
I think the newest thing that has come out recently in science, which I found to be really interesting, because I get a lot of questions from beekeepers about the reproduction of this mite, right? So, and you did a great job showing how it changes versus them doing worker parasitism of a cell versus like a drone cell, and the different life cycle and how it coincides. But what's now fascinating is there's work coming out showing that there could be multiple mites reproducing in the same cell. So now you've got this kind of aggregate population, and you talked about that, which is interesting. So it definitely makes a lot more questions now for us trying to figure out how to manage this thing if there are multiple individuals developing inside of the cell, instead of kind of this Game of Thrones mentality where the brother mates with all the sisters and you've got all this creepy stuff happening. One mite is supposed to be the survivor, one or two. But yeah, I think as far as going outside of the brood cells, I mean, we definitely don't see Varroa mites hanging out if they're not attached to bees or they're not in a brood cell. They're very rarely hanging out on equipment. So I imagine this transfer is really quick.
All right, one more question and then we should probably move on.
Should you test a new package? That's a good one.
Yes.
All right, two more questions and then we'll move on.
That's it. Yes, that's one we're going to be covering next week and next webinar, right, Kim?
Yeah, we'll talk about packages versus nukes versus pole colonies, and let's see. Is there a benefit to a brood break if the mites feed on bees outside the cells and still pass viruses that way?
Yes.
She just wants to say yes. Yes. Moving on. Okay,
yes,
what's next?
Yes, there is a benefit. And so we're going to actually talk a little bit about population and colony growth right now. So
perfect. Yeah.
Look at this. These questions are perfect. Perfect lead-ins.
There we go. All right.
They're paying attention. Ooh, I love it.
Yeah, they are.
And we have a couple more times with question breaks, so continue putting your questions in there, and hopefully we'll get to them all either live or we can do written ones too. So, all right. So let's move on.
Next slide. There we go. So, we were just talking about kind of brood breaks and population breaks, but it's important to understand how a colony grows throughout the season first. So what we have in this yellow line here is the bees are increasing and your mites are increasing kind of at the same rate, but delayed by a bit of time. And so, Yana, can you hit the thing one more time? I think there's a star that comes up. There it is. Yeah, so what you can see here is that your bees are increasing, your mites are increasing, they're kind of keeping pace. And then as your bees are decreasing, getting ready for winter because they do not overwinter at 40,000 bees, they start decreasing their numbers down to about 10. Your mites are still increasing, so there's a lag in that increase. And it's really important how these numbers switch kind of very quickly. So you go from a 6% infestation to a 35% infestation. Now, both of those are way too high, and you should never be up that high, but you can see how just looking at the numbers, that can happen very quickly. And where that star is on the map is about the time when your bees are making primarily their winter bees. Actually, it's a little bit over towards August, a little bit more. And so this is when they're starting to make their winter bees. So your bees have to be the healthiest they can possibly be at that point. Those are the bees that have to live for 5 to 6 months. And so you can't just go at this point and say, I'm going to do a treatment and knock those mites down, because the bees that are being produced there actually were about a month ahead of that. So you're looking at June, July as making sure that you have your mite populations under control, so that when you start making your winter bees, that they're as healthy as they can possibly get. So making sure that you're sampling and looking for mites throughout the season, throughout this full season, is really important, not just working on a schedule. So this whole entire graph can actually shift as a result of poor forage. So what happens is when there's poor forage out there, the bees will increase population. They hit a dearth, that's poor forage, and they'll level out. They won't continue increasing. So they level out their reproduction. And during that time, they still have brood, they still have mites producing. Those mites can actually increase at a faster rate when those bees are kind of leveling off, or I guess it's the way the numbers work. So they're not really increasing, they're just, those numbers are switching. And so you can actually shift this whole entire thing a month or 2 months in it, you know, further to the left, where you could see your mite population's getting out of control much sooner than you normally would if we have a particularly dry spring, summer, and there's no forage out there for the bees. I hope that all makes sense. So next slide. And so one of the questions that I get most often is, how do I know if my hive died from Varroa? So Kim and I do a lot of hive autopsies, tons of them, because we think it's a really important learning experience for pretty much everybody involved. I really like looking through, well, I don't enjoy looking through dead outs, but I know it's important, so we do it. So next slide. So you want to kind of let the time of year help start to guide you on what you're seeing in that hive. So remember those numbers that we saw at the beginning of the presentation, that during the summer, queen, brood, and food issues is what we see as the #1 cause, or the beekeepers are reporting as the responding as the #1 cause of death. In the fall, it's Varroa mite and their viral complexes. In winter, it's Varroa mites, viruses, bad winter, or starvation. And so you want to think back, when was the last time, if your hive died during the winter, when was the last time you saw it flying? Because early fall deaths can be different than a full winter death, and the way that they present would be different. So you want to think about, when was the last time you saw the hive active. Next slide. So you want to rule out a couple things first. You want to make sure there was still plenty of honey in the hive. So up at the top here, that's obviously a live hive, but you can see the honey in the corners. You have brood. Down in the bottom, you can see that's a dead out hive. There's no honey anywhere in this hive left over, and there's a big dead cluster right on the front of that frame. So inadequate stores could mean starvation. So you want to rule out starvation. Next slide. You have no signs of queen issues. So first of all, there's lots of honey left. You have no signs of queen issues. And queen issues would be no queen in the dead bees, no eggs or brood, which if it's dead, you wouldn't expect to see very much of that anyway. Lots of drones. So you can see in the dead bees, you would have tons and tons of drones in there, many more than you would expect. Many emergency queens, cups or cells. That didn't seem to work out. So the top picture here is a healthy hive with the queen and of course her attendants and bees. And then in the bottom, you can see that brood pattern is very drony, so it's very lumpy. If you go to the next slide, you can see these are all signs of a queen issue in that hive. So you can see lumpy drone brood where there should be worker brood, empty queen cups, and drawn out queen cells that didn't seem to make it. So there's no signs of queen issues. Next slide. So if the last time you saw it alive was fall or early winter, and what you would see was only a handful of bees, brood, and the queen remaining in what was once a strong hive. So what happens in this case is that the viruses have gotten a hold of the bees, or taken hold inside the bee colony, and it makes the adult bees feel sick. If the weather's still warm, what happens is those adult bees will fly out of the hive and go die someplace else. They feel sick and they don't want to spread it to the remainder of their colony. They're much nicer than I am. When I'm sick, I end up going to work and spreading it to everybody else in my office, unfortunately. And so what happens over time, as these bees are leaving and dying someplace else, that cluster size decreases until it's too small to keep warm during the winter. So this picture here is a hive that was very strong going into the winter or into the fall, and it died right around Thanksgiving. Thanksgiving, and that was all that was left of dead bees. It was about a cup and a half, and it was strong before that. So next slide. And so one more, keep going. So as you can see, when we look closer at this bottom board, all those red arrows are pointing at Varroa mites that were mixed in with these dead bees. And so obviously there was a Varroa mite issue with this one. Next slide. And so this can be problematic in those early spring, late fall, early winter deaths because of this mite bomb effect that we've kind of coined over the last couple of years. So what happens is these bees that are infected with mites and feel sick will leave the hive until the hive kind of is very, very small. If the queen dies before all of the worker bees die, the remaining workers will actually go and spread to other hives, bringing mites with them. So this researcher a couple years ago, what she did was she found a hive that was dying from Varroa mite, was collapsing from Varroa mite, and she marked every bee in that hive. She let it collapse, and then she sampled every hive for a 2-mile radius and found marked bees in every single hive. So what's happening is these bees are spreading out, leaving the hive with their mites and joining other hives in the area. And this can look very much like absconding. You'll find a couple handfuls of bees in the bottom of the hive, or maybe even less than that. So in the fall, I get quite a few calls that say my hive is absconded, when in fact it was a mite-related death and the bees left and took residence in other hives. So next slide. So if you have a late winter death, you would see lots of dead bees on the bottom board, a small dead cluster on the face of the frame. So what happens is the bees who are not living as long as they should be living, the 5 or 6 months that we need them to, well, maybe not 6 months, but 5 months that we need them to over winter, especially if they have high mites, high viruses, their immune systems are compromised, they're dying sooner than they should, and they're dying over time. And so what happens is they die over time and then end up with a full bottom board. Oh, keep going, go back one more. You end up with a full bottom board. Yep, there we go. A full bottom board full of dead bees. And so you want to look for mites on that dead cluster or on the bottom board. So you can actually take a scoop of the bees from the bottom board and do a mite roll on them and see what you get for numbers. So next slide. Or you can look at the cluster as well. So each arrow here is pointing to a Varroa mite on that cluster, and that was just observing them right up on the front, just looking at the tops of the bees. So there's probably a lot more in the cells or in the bees in the cells. Next slide. And so you also want to look at the dead adult bees and make sure, and you'll probably see some abnormal deformities. You'll see things like shortened abdomen, stubby wings. There'll be hairless bees, greasy bees, black bees. These are all signs of viruses. We looked at those pictures earlier, and many of these viruses are associated with high Varroa level. So if you're seeing lots of deformities in your bees, there's a good likelihood that you actually had a mite problem. So sometimes we'll have a hive that we do an alcohol roll on, a dead hive where we do alcohol roll on the bottom board, and we actually don't get very many Varroa mites in that. And but it feels like a Varroa mite dead hive if it just feels like that's what caused the death. And when we look at the bees, we'll actually see those deformities. So what happened is that a beekeeper did a treatment, took care of the Varroa mites, killed them all off, but the viruses had actually still taken hold and ran amok in that hive and ultimately caused its death. So the treatment was too late in that case. So next slide. You also can see white crystals stuck to the cell walls of the empty comb. So these little white crystals are actually mite poop, and you'll see a lot of that in a hive that has really high mite levels. Next frame. Next slide. Sorry, this is not mite poop. This is normal crystallization and fermentation. I had a beekeeper send me a picture like this a couple weeks ago and was like, I had such terrible mites. I was like, no, that's actually normal. So that's not mites, not mite poop. So next slide. Definitely
more delicious than mite poop for sure.
That is more delicious than mite poop, yeah.
Crystallized honey.
Yep. And then the remaining brood looks kind of sick. You got a spotty brood pattern. That's really common with any hive collapse. So you want to make sure that that weird, sick-looking brood is not due to something like American foul brood, chalk brood, European foul brood, anything like that. So you want to make sure that you're checking for those symptoms. A normal dead brood, so one that's not dead from a brood disease, is gray-white in color. So it's like a goopy gray-white, and you don't have any roping. So see in that top picture with American foul brood, that roping, that's a toothpick that was put into that larvae, twirled, and then pulled up, and it causes it to kind of extend out, spread out, and that's called roping. You do not see that in a normal dead brood or a Varroa dead brood. And there's really no terrible smell. So there's sometimes a bad smell, but not a terrible one. It doesn't smell like fish or rotting meat, which is important. So next. So healthy brood, if you were just looking at your hives during the summer, this is what this healthy brood would look like. It would be uniform in appearance, few interruptions in pattern, pearly white. So you can see in the top left picture that pearly white larvae shaped like a C. You could just scoop that out, pop it right in your mouth, and it looks like it would taste delicious. Nice brood pattern. Cappings are convex, not perforated or greasy, and there's no offensive odor. So next slide. So this is a hive that was starting to collapse from Varroa Mite. So you can see how they're starting to get skips in the pattern. Next slide. And then as you look closely, you can see, oh, I think I lost Kim, the arrows in this one. You can see that there are cappings that are open with brood in them and then there's some cappings in the top left-hand corner that have perforations in them that are very distinctive signs of Varroa Mite damage. So next slide. Oh, there they are. Got them. So there's the arrows. So you can see these arrows are pointing at brood that's open, brood that has perforations in it, or cappings that have perforations in them, and those are signs, early signs of the Varroa Mite collapse, hive that's collapsing from Varroa Mite. So next slide.
I always tell my beekeepers as an interjection, if you pull out a frame of brood and you see their little faces looking at you in the immature phase, that does not mean they are coming out. That means they are dead. So you should never see them uncapped once they're capped. They go from larva to cap to adult bee. So if you're seeing those little faces, it's not a good sign.
Yep.
Sorry, not too simple.
No, absolutely. So next slide. So again, here's some more signs of that. So in the top left-hand picture, you see some adult bees that kind of started to maybe chew out a little bit. They've been uncapped. They're dead underneath the cappings. Same thing in the bottom. You have these cappings that are the bottom picture. You have these cappings that are exposed, very spotty pattern. And then up in the right-hand corner, we have an interesting situation. So often when a hive is collapsing from Varroa Mite, you'll find that they start chewing up the dead pupa and pulling them out. And so here right in the middle of that picture, you can see a couple of cells where they've started chewing up that pupa and pulling them out of the hive to dispose of them because they died. So next slide. And so when you remove dead bees, you will find Varroa on the brood and the bees that are removed from the cells. So this top left-hand picture, you can see the Varroa Mite right on the back of that adult bee, down on that pupa that was pulled out in the bottom left. In the cells at the top right, you can see the Varroa Mites right in the bottom, and then off this dead bee that was pulled out of this hive in the bottom right picture. So next slide, and we'll take some questions.
Okay, so all right, do you want to take your favorite one, Kim, or shall I just choose one here? So actually, maybe I'll jump in with this one. Can black queen cell virus be transmitted by Varroa?
Sorry, I was trying to get my, okay, there we go. Yes, so black queen cell virus, we used to see this probably as one of the most common viruses to show up in samples when we submitted them to the labs for analysis, but we're not seeing it nearly as common as it used to be. Although when we do get reports of black queen cell virus, we don't tend to have beekeepers worry as much, especially if they're not visual symptoms of black queen cell. But yeah, it is definitely one of the viruses, and correct me if I'm wrong, Jen, that has been shown to be transmitted by Varroa mites.
I believe so, yes.
All right, here's actually an interesting question. Scott Herbert asked, just to clarify, these images are pretty extreme cases of Varroa infestation, correct?
Well,
So, yeah, it's not really.
I wish we were showing you kind of the massive pileup accident on the side of the road that, and put on your seatbelt kind of thing, but to prevent that. But the reality is, especially some of the frames that Jen showed where you just have a couple of uncapped pupil cells and you can see, the eyes and the head, those tend to be sort of the initial stages of when a hive is really going down the road of having real high Varroa levels. So we see that way more often than we would like. They can definitely come back from that, but when you start seeing some of the pictures that she showed where they're completely wiped out, there's a ton of them on a single frame, you pretty much leading to collapse. But yeah, and I don't want to take away, Jen, because you probably got more information here on this one.
No, that's absolutely it. I wish we saw it less frequently than we did, but the progression, right? Like Kim said, it starts off with a couple uncapped pupa, then start removing, eating those pupa, and then it's a quick collapse after that, where you just see lots of really spotty brood, lots of dead brood, and then it's goner.
Yeah. There's a couple of really good ones on here. Let's do this one. Can you reuse frames from a dead out?
Yes. The only time that I say do not use frames is if you have American foul brood. And that's for both of our states, that's a burning situation. But yeah, especially when it comes to, so with the caveat, we don't really understand yet how long viruses are staying on comb after death due to Varroa mites and viruses. So the Varroa mites, most likely, if your hive dies during the winter, the Varroa mites die along with it. They also freeze to death eventually. But the viruses, we don't know how long that stays on the comb. There is some research going on out of several different places, but probably the one that's closest to where we are now is out of Cornell. So Emma Mullen there has been doing some research looking at longevity of viruses on comb. So hopefully stay tuned. There'll be more information coming out soon on that.
Yeah. This is another good one. Is it okay to let your hive be robbed out that had parasitic mite syndrome?
No.
That's a famous one answer question from Jen. No. So yeah, it's a good question though. Yeah.
Because we don't know how long those viruses stay within the system, it's better to be safe than sorry. If you have a hive that collapses for some reason, don't let it get robbed out by another hive. Figure out what the reason was, and then go from there.
Yeah.
We typically find that the honey is less of an issue as far as transferring viruses, but the bee bread definitely can be a place where they are picking up viruses.
Yeah. There's a couple of questions that we're going to go in great detail, so I don't want folks to feel like we're glazing over it because it's a big topic, but when should I treat for mites if my bees are coming out of winter? When should I treat for mites if I have a new package? What are some of the treatments to use? For instance, is this treatment oxalic acid better than another treatment? There's some really great questions on here, and I can already tell our treatment episode is going to be fantastic. I'm excited. This will be a great conversation because I think there's a lot of really good options here that people can use, and yeah, I'm excited.
Yep, and also we'll be answering a bunch of these questions offline as well. So there'll be a document that goes with this where we answer some of these in a little more detail and can finish up any of them that we missed this time around.
Yeah. Hopefully with the, oh, sorry.
No, go ahead.
I was gonna say, hopefully with the series of webinars, we have plenty of time for your questions. So even if you don't get the questions answered today, if you write them down or jot them down, you'll have other options to present them again. So, yeah.
Great. All right, so let's move on to the last section, and we have some more Q&A time too, and so let's move on to the last section, which is where you can go for more information. So.
Oh yes, yeah. So go ahead, next slide. So there's lots of places where you can get more information about Varroa mites and viruses, and so if you're not from actually, if you're anywhere in the United States or even Canada listening to this right now or watching this, you're more than likely have an apiary inspector or an apiary program of some sort, or a research station that's doing some work with bees that you can reach out to. And so we actually have a organization called Apiary Inspectors of America that both Kim and I are members of. Actually, Kim is the president of. And you can go there and find out. There's a circle around. There's a website. There's a circle around where you can find who your apiary inspector is. And I promise you, all of us love talking about bees. It's a little bit of a problem. We get boring very quickly at house parties. And that's probably the best person to reach out to in your area. So what we're describing here is what we see in New England. That could be very different than what you would see in Florida or in Alberta. So finding out what's going on in your specific area is really important. So please reach out to the apiary, your local apiary inspector. They love talking about bees. Next slide. There's also a really good organization called the Honeybee Health Coalition. They have a free best management practices, or a bunch of free documents. Number one is the tools for Varroa management. So they have I think it's up to 30 pages now, 40 pages. It's a very long document, but it really boils down all the new research, everything that we know about Varroa mites and treatments and all of that into one really easy to read document. And so if you just type in their address, Honeybee Health, or type into Google Honeybee Health Coalition, it'll come up with their site, and it's a free document. They also have this really neat decision-making tool with an app that you can put in the temperature outside, the time of year, what your bees are doing, and it'll give you the different treatment options that you have at that time. So it's a really useful website and useful information. They also have a best management practices for health document as well, and I think that one runs about 60 or 70 pages, but it goes into a lot of detail about all the best management practices that you can do in your hive. So next. And then there's also the Bee Informed Partnership. So they do a lot with citizen scientists. They ask beekeepers to participate and offer data, and then they make these beautiful maps and that are completely available for people to use. So they do an annual loss and management survey. They also track they have hive monitors out there. So hives that they have monitoring devices in them. They look at weight, temperature, humidity. They also run the Sentinel Apiary program where beekeepers take samples for Varroa and Nosema, and then they produce maps for them so you can see what's going on at that area. And then they also run the Mite Check website, which is a website where you can self-report your mite levels. So every time you do an alcohol roll or a sugar shake, or monitoring in some way, you should be inputting that, or you could input that data into the Mite Check website, and that would help other beekeepers in the area figure out what's going on. They also house the data for the APHIS National Honeybee Disease Survey. I did see a question, somebody asked how many states participate in that. And actually, I don't know. Kim, do you know offhand how many states are participating in the National Honeybee Health Survey?
I think this past year might have been like a record. It was like 44 or something. I feel like
yeah, I thought it was somewhere around 40. So yeah, that's about right. And so what that program is it's a federally funded through APHIS, where a lot of the apiary inspectors in their respective states go out and work with beekeepers and then submit data, and they look for viruses, pesticide loads, nosema, and Varroa mite in those hives. And those are some really important data for each of our states, because it's some of the stuff we don't normally get data on. And so this is a really good site, and you can explore the maps in many different ways. And they have historical data up there too, so you can see what happened over past years. So next slide. And then going to your local, state, national honeybee organizations and their meetings. So going to meetings can be really important. The national and state meetings will often have researchers that come and talk. And that's how that's how you figure out what's going on and hear from kind of the experts on these things. And then you can also learn about and meet other different types of beekeepers. We often get stuck in beekeeping ruts where we do the same thing year after year after year. And sometimes at these national meetings, you can find out what's going on in other regions in the country and see if that will adapt, or you can adapt that to your management style. And then your local meetings are a really good place to find out how to keep bees in the area. They're going to know the best methods for that area, that climatic zone or those microclimates. But just remember that not everything that somebody tells you to do may work for you. Not necessarily wrong, but I like to say that if you ask 5 beekeepers the same question, you'll get 17 different answers. And often within one sentence, you'll have completely opposing answers that they are adamant that both of them are correct. And so making sure that, if you try something with the understanding that it may not work for you, even if it works for another beekeeper. And then also often in these groups, there are hundreds of years of experience in the room. I have several beekeepers in the state that have been keeping bees for more than 50, 60, 70 years, and they're still doing it. And they've tried a lot of things over the years. They've seen it change, and they're a really good source of advice and information. And then also getting a mentor and working with other beekeepers can be really important. So it gives you a chance to look in many different hives. So one of the quickest ways to learn beekeeping is to work in many, many, many different types of hives. And so getting a mentor and working with other beekeepers in your area is a good way to do that and not spend a lot of money. Also, working with other beekeepers in your area, it's important for pest management. So when it comes to Varroa, remember we talked about those mite bombs where those mites are dispersing in those sick hives up to 2 miles away from the original hive. If you're working with everybody else in the area, or the beekeepers are all in communication, that can really help, because if you see a problem in your hives, you can communicate that to other people, and then they can look out in their hives. So in Maine, we have a lot of communities that do work together, and they're pretty effective at keeping their Varroa throughout the area low, and have less impact on each other's hives because of it. So next slide. And then, so for a shameless plug, EAS, the Eastern Apicultural Society, will be hosting, Maine will be hosting the Eastern Apicultural Society meeting in 2020 up in Orono, Maine. It's the first week of August. And we're specifically doing the art and science of beekeeping. That's our tagline. And we're going to be talking to we've invited some of the leading researchers on Varroa Mite to give talks during that conference, because we think it's really important. And both Kim and I will be there talking about Varroa Mite, along with several of our other colleagues that are apiary inspectors across the country. So please come and join us. And then next, you want to do this one, Kim?
Oh, yeah. So another shameless plug. Thank you very much. So we have been doing a live version of these webinars in a sense that it's a day-long training for beekeepers, and it's been really awesome to watch. We've been doing these through MDAR in Massachusetts, through using UMass Extension folks as our big collaborators on these, specifically Anna Whitehead at UMass Extension is the honeybee educator. She just joined me, and more importantly, the beekeepers we're sort of just the stewards of the day, the facilitators, so to speak, but the beekeeper participants that sign up for these day-long workshops really do interactive and hands-on the entire day. And the learning that happens, the communication and the sharing, it's just really awesome. It's so powerful. And if you have an opportunity to come to these, I highly recommend it. Again, a shameless plug. There is a fee for these. I think it's $40 or $50. I'm not sure what the cost is, but it will cover the kit that you can see in the corner. So you'll get your own Varroa Mite kit with everything that you need. And then also we put in a little bit of protective equipment for some of the mite treatments that you can use as a beekeeper. And then also inside the kit, you'll get a ton of handout materials that you can take home. And probably the thing that I think people have enjoyed, kind of the fun take home, is the life-size Varroa Mite that we've got a couple photos of that you can take home and share with your family. So hopefully our goal, right, is for that to be the only Varroa Mite that you have and are happy about seeing is that one on the card.
The next slide.
Yeah.
Yep.
So these are some of the folks at the workshop. On the next slide, you'll see having fun with these hand-cut Varroa Mites that we made. We made these actually to scale. So, oh, here we go. Oh, $40. Oh, that's a steal. That's a steal. If you want to see the location, oh, maybe that's on the next. If we go down, oh, there we go. Oh, I also forgot to mention, the beekeeper in this photo you get a free t-shirt. Who does not love a bee t-shirt? Hello, I need more of those. So you get a free t-shirt designed by Hannah Whitehead that says "Fight the Mite" with a little boxer bee on it. And we've also been graciously had apiary diagnostic kits donated from a bee supply company to hand out during these workshops as well, donated to UMass for this. So the goal of these is that we go through basically the webinar idea that we've been doing with the Northeastern IPM Center was born out of an interest in the initial webinar combined with the amazing popularity of these day-long workshops. And so we've kind of synthesized that and turned it into a webinar. But if you're interested in the live version, I highly recommend these. So please join us.
You've missed your calling for sales, Kim.
I know. They're so much fun. I mean, look at these people. They loved it. Look at this. So, Jen chose the photo with my face in there, so I apologize. But you can see, we also have our Massachusetts honey ambassador, Ryan Dugan, who's now a student at UMass. I'm giving a shout out to you, Ryan, who joined us as well. So, yeah, all types of folks come out to these, all different levels of beekeeping experience and backgrounds and education. And I think they leave at the end of the day, not only with, hopefully making some fun friends and bee mentors and bee friends in general, but a ton of information and materials. One of the things that tends to be the most impactful from these workshops is that people get a case study, which is basically a scenario from one of our beekeepers or experiences that we've had in Massachusetts. We don't obviously have names on it, but just in general, beekeeper X, and this is what they're doing. And you have to create an entire Varroa Mite plan for this beekeeper and also then demonstrate as a team how you would apply the different treatments using all the correct materials, protective equipment, the actual beekeeping equipment itself, if you need to remove parts or add things or whatever. So people really, really like that hands-on piece. So yeah, again, sorry, I can't talk about this enough, but they've been really popular. So yeah, join us.
Right. So before we go to some more questions, there's a couple of things I want to cover. We have 2 more webinars coming up. The next one is on mite monitoring and treatment, and a lot of the questions that came up today will get answered in that webinar. And then we have creating a Varroa Mite IPM plan on Monday, April 6th. And a live demonstration and Q&A on April 20th. We're going to actually be going to Massachusetts and recording some footage to demonstrate all of these things that we're going to be talking about in the next month. And same, we'll have Q&A interspersed in there. If you're registered for this webinar, you will be registered for all of the other webinars. and they are all going to be recorded. So same as this week, in about a week, you should see a copy of the recordings for each one. So if you have a time conflict for one of them, don't worry about it. It will be recorded, and you will get, if you're registered, you will get an email from me with a link to the recordings. So, all right. So we're going to just ask some questions, some polling questions, and then we'll go back to answering some more Q&A until the end of the half hour. So hopefully you should see the poll questions in front of you. I know some people had some problems earlier seeing the poll questions, and I'll just be quiet for a few minutes while you can answer those. I think there's 6 or 7 questions this time, so it'll take a little bit longer to go through. There are no dumb answers. It is totally stress-free. Just have fun with it and guess if you have no clue. So totally fine. All right, we got to 60%. So I'm going to share the results. And so after this webinar, more people feel moderately knowledgeable. That's very cool. We maybe can move you up the scale here. So you're going to be in the extremely knowledgeable category by the end of the four webinars. So, and then I'm going to ask Jen or Kim to go over the multiple choice questions or the questions that involve content, so that I don't mouse it up. So.
Oh, sorry, I got to move. Okay, there we go. My light just went off. I was sitting still for too long. So common signs of mite damage include open and damaged pupal cells, check. Holes in pupal cappings, check. Odor coming from the hive, check. Emerging adult, yeah, it's kind of, probably not so much as other things. Emerging adult bees with deformed or missing wings, check. And then visible mites on brood and bees, check. Good job. Yeah, so I would say odor coming from hives is it's not a common sign, but occasionally you'll see that happening. So great. And then the other ones are just for us.
Okay, all right, great.
Currently monitoring Varroa mites in your hive, yes, we like that. Which methods do you currently use to monitor mites? Alcohol roll, visual inspection are the number tied for first. And then if you don't currently monitor, why not? Other reasons.
Other. Oh.
Other. All right. Of the 83% that's other, we'll have to come up with some other multiple choice options other than the ones we have. Or maybe we can see if there's a way that we can ask you what those might be, since it's a sizable number that was none of the above. So, all right.
Hoping they just need a mite wash jar. We can solve that problem.
Yeah. And then there's a wide variety of different treatments being used. So, great.
All right, lovely. Okay, so we have time for a few more questions before we need to close up. So let me see if I can make it go back. There we go. And Jen and Kim, do you want to choose some of your favorite questions that you'd like to answer that you?
Yeah. We have one here from Roger. What are different states and the USDA doing to monitor mite counts on nukes and packaged bees being shipped into their states? Has a certification program ever been implemented or discussed?
So there are states that have requirements that bees that packages are shook from have to be treated for Varroa mites prior to being used for package shipments or preparation. However, that's super variable depending on the state. We've had a lot of beekeepers actually in Massachusetts have interest in this. And unfortunately, so a couple things come up because Jen and I are both in a regulatory position in our states. Obviously, we are not able to enforce regulations on other states to, treat for mites or force beekeepers in those states to treat for mites. We also are, we also cannot make sure that bees have been treated in the sense that we are not registered pesticide applicators, nor are we able to enforce pesticide use on folks who manage bees. We can provide recommendations and we can encourage people to use certain things, but unfortunately we can't say, all these bees need to be treated and everybody needs to treat, every single bee that comes into the state. But we can definitely have some regulatory capabilities to help mitigate if diseases are a problem. It would be really interesting if the USDA would come up with a larger program that could be addressed at the national level. It would help alleviate issues with our states trying to figure out how to do this. But then again, I think we also would run into some interesting situations where we could have resistance to some of the chemicals being used. So yeah, we are working more diligent with the Apiary Inspectors of America group to talk about our state requirements and how we can be a little bit more uniform, and not only in how we provide inspections for health certificates as bees move around states, but also as we look at emerging threats and obviously existing threats like the Varroa Mite issue that seems to be celebrating its 30th birthday here, and a little bit more than that in the U.S. So yeah, Jen, you might have other things to say. I didn't mean to take over that question.
I was just going to mention that there's no kind of standardization across all the states or at the federal level, mostly because when Varroa Mite first came in, or was first introduced into the United States, there were some regulatory, there were some things that came up and at the federal level, and they did quarantine hives and all of that, but it's so widespread that we just at this point assume that every hive has Varroa mites in it.
All right, and we're kind of running out of time, so
okay.
So we can, and a lot of these questions that have not been answered, we will, I think, answer it next week or in the coming weeks. And but if there's something that won't be answered, that I think Jen and Kim are willing to type up some answers that we can share. I also wanted, Roxy had asked for the live demonstration. If you're in close proximity, can you attend in person? And I can talk to Jen and Kim, but I think probably not, because we're going to be doing video recording, and it's so incredibly boring to watch that, having done it.
It's like 10 hours for 5 minutes,
right,
something like
right.
that.
Yeah, so it's not really designed for having people. But I think if you're in close proximity, go to the workshop that Kim was doing such a great job in promoting, and if you haven't already. And okay, and I want to cover a couple of quick things before we end. The Northeastern IPM program, one of our prime goals is to increase communication between Northeastern states around IPM. And we love it that Jen and Kim applied for a grant and from different states to work on this project together. And one of the ways that we can facilitate that is by putting colleagues in touch with one another. So we have this place on our website called Find a Colleague, where you can list yourself and your interests, and then if you're interested in collaborating on projects, you can find other people who are also interested in, you know, in your case, in honeybees. So that's one thing I'd like to share. Today's webinar is going to be on-demand recording. It'll be available in about a week, and I will send you an email with the link once it's ready, and you'll be able to watch it as many times as you like and share it with colleagues. It's going to be freely available. And we just want to say thank you to all of the organizations that provide funding for us, the USDA NIFR for the Northeastern IPM Center, the Massachusetts Department of Agricultural Resources, and the Maine Department of Agriculture, Conservation and Forestry. None of this would happen without tax-supported funding and grant funding for these projects. So thank you for what you're doing this time of year, getting your taxes ready. So thanks for joining us. I hope it was really helpful. It was very interesting for me, and I hope it was equally interesting for you, and look forward to seeing you in a couple of weeks. And Jennifer and Kim, thank you. It was always fun and interesting. Is there anything you want to say before we end?
Nope, thank you.
Yeah, it looks like we had a syntax issue, an error on our end with question 5. We're getting a lot of feedback for that. So I think the other may have been our issue. So don't fret. We see you and hear you. We're going to make sure those are not the cases in the future. But thank you so much, everybody. We look forward to hanging out with y'all again. And thanks for listening to us rant along about bees and mites. And if you're interested, come back again.
Terrific. All right. Goodbye.
Bye-bye.